Okay, students, so the second topic I'm covering here is classification of various enzymes. Okay, we have already understood about the nomenclature of enzyme. If you have not gone through that, so please go through that. I will provide the details into the description also. Okay, so if we are starting about the classifications, so classifications can be categorized into the 6 categories. Okay, so the first is oxidoreductase. Oxidoreductase included those enzymes which are involving in both the reactions, means which are involving in oxidation as well as reduction reactions. The examples are oxidase, reductase and dehydrogenase. Okay, so the oxidase enzyme means those enzymes which are involved into the oxidation reaction. If we are talking about reductase, so reductase are those enzymes which are involved into the reduction reactions, and dehydrogenase which is involved into the removal of the hydrogen. Dehydrogen means removal of hydrogen. Okay, so those enzymes are mainly undergone through the category oxidoreductase. Okay, if we take in the example, so first one is example is ethanol. Here in the ethanol, the two hydrogens are removed, one from here and second one is from here. So the nicotinic adenine diphosphate is converting into the nicotinic adenine diphosphate with the hydrogen, and two hydrogens are added here, and ethanol is converting by the removal of hydrogen into the form of acetaldehyde. So the alcohol dehydrogenase, because here is the removal from the hydrogen ion from the alcohol, that's why it is known as alcohol dehydrogenase, removal of the hydrogen from alcohol. Okay, so ethanol is converting into the acetaldehyde. Now the second one is transferase. Transferase means those which include the transfer of various functional group. Okay, it may be methyl, it may be glycosyl, it may be various groups. Okay, so it means from one substrate the group is transferred from one to another substrate. Okay, if we are taking example, so here is the carbamyl phosphate. The carbamyl phosphate is involved carbamyl group as well as phosphate group. So I have mentioned here the carbamyl group is removed from here and it is combining with the aspartate group. So the final compound you will get is carbamyl aspartate, because the carbamyl group is removed from here and it is attaching with the aspartate. So from first substrate it is removed and in the second substrate it is attaching. Okay, so you will get the result as a carbamyl aspartate. It is known as transferase because from one substrate it transfer from to the another substrate. That's why it is known as transferase enzyme. The third one is hydrolysis. Okay, hydrolysis means in this type of reaction or these enzymes are responsible for reactions which involve removal or breakdown of water. As I have already told you, where you have seen the lyase is mainly the removal or breakdown. So hydrolysis means removal of water molecule. That's why those enzyme which is removal for the breakdown and removal of water is known as hydrolase. Example, if the sucrose is reacted with water molecule with the presence of sucrase. Okay, sucrose here is the substrate and the enzymes are named as per the substrate. So that's why the enzyme are sucrase. So when the sucrose is reacted with water molecule, it will be converted into the glucose and fructose and the water molecule will be removed. Okay, so that is the example of hydrolysis. Now the lyase. Lyase means the removal of functional group. I have already told you where you have seen the lyase word. So lyase word is basically indicated the removal and breakdown. So the examples are decarboxylases. Decarboxylase means which is remove the carboxyl group, aldolases which remove the aldehyde group, and synthesis which is involved in various synthesis while it included the removal of various functional group. Okay, if we take the example, so here is the example. Suppose we are taking the pyruvate. Okay, so the pyruvate, with the presence of pyruvate decarboxylase. Now here is the decarboxylase, means removal of carboxyl group. So the pyruvate with the removal of carboxyl group will be converted into the acetaldehyde. Okay, and the enzymes which is involved in it is pyruvate decarboxylase. Now isomerase, the fifth one is isomerase. As per the name indicated, it is involved in formation of various isomers and epimers. Okay, as it is involved into the formation of isomer, it means it is helpful into the rearrangement of the various molecular structure. Okay, so as you all know that glucose 6 glucose and fructose are isomers of each other. So when the glucose 6 phosphate is reacted with phosphoglucoisomerase, okay, it will convert it into its isomer form, into the fructose form. So when you will see the isomerase in any biochemical reaction, it means this enzyme will be definitely form the isomer of that particular substrate. Now the sixth one is ligases. Okay, ligases and lyases both are different. Lyase is for breakdown and ligases is for synthesis. Okay, so it is involved in various synthesis, various bonds. Okay, so examples are synthetase because it is involved in synthesis, and carboxylase ASE. Okay, carboxylase because it is involved into the formation of carboxyl group. Okay, so synthesis of various bonds are mainly due to the ligases. Examples are when the pyruvate is converted into the when the pyruvate is reacted with the carbon dioxide and one molecule of ATP with the presence of pyruvate carboxylase. Okay, here is the carboxylase, so it is the ligase enzyme. So due to the bond formation, the pyruvate will be converted into the oxaloacetate, and one ATP molecules will be consumed and it will be converted. The adenine triphosphate will be converted into adenine diphosphate with along with the phosphorus group. Okay, so due to the rearrangement of pyruvate, it will be converted into the oxaloacetate. Okay, students, hope you all understood about the classification of enzymes. Okay, in the next topic I will cover the enzyme inhibitor. Thank you.